Hey guys, welcome back to our second set of notes for chapter 5 here. We left off talking about enzymes, and now we're going to jump into a little more information about the reactions that are actually occurring in our bodies. So let's get right to it here. Molecules interact in organized pathways that we call metabolism, and metabolism is really just the sum of all chemical reactions that occur in your body. These activities that the cells acquire and use energy are our metabolism, and a metabolic pathway is a series of enzyme-mediated reactions. So in the video I showed you one little enzyme with one little substrate, and it performed one reaction. And that's a nice foundation, but really when you look at the reactions occurring in you and I, it's more than one enzyme. It's many enzymes working in a chain or working in a loop, and their products go on to be, the reactants in the next piece of the chain. And so they're a little more complex in the real world. So cells are going to build molecules up, rearrange molecules, or break down these organic substances. And these linear pathways can run from reactant all the way to product. A lot of reactions are cyclic in nature that are occurring in us and in plants, and they are regenerating a molecule that is then used in the first step of the reaction and will continue on to do the whole reaction again. So controlling metabolism has a lot to do with concentrations of reactants or products. They can actually make a reaction slow down as you make more product, or even reverse the reaction if you make too much product. And so we have mechanisms that can adjust our enzyme production, or activate enzymes, or slow down, or stop, or inhibit our enzymes. And so that's called feedback inhibition. Feedback inhibition decreases or stops the enzyme activity, and it's a process where the product of the reaction slows down the other enzymes in the chain so that we don't make more product than we need. We also have some regulatory molecules that can activate or inhibit our enzymes. Some of these molecules can bind to the active site so no substrates can get to the enzyme. Others can bind outside of the active site in what's called allosteric regulation or allosteric inhibition. And when they bind to the back of that enzyme, it will keep the enzyme from being able to actually do something with that substrate. So there are a lot of ways to just shut these systems down or slow these systems down, because while metabolism is good and it's incredibly useful, it's just like anything. It's under the same restraints of homeostasis that all of our other systems are, and it can't just run wildly out of check. We want a reaction to go until we don't need it anymore. Then we need an off switch. That's where allosteric inhibition comes in, and that's where inhibitory cofactors and coenzymes come in. So this is the example of a linear reaction. And so down here's our product. It's where we want to get to. And we start off with our reactant. It goes through an enzyme and makes an intermediate molecule. That goes to a different enzyme. It makes another intermediate molecule, which goes to another enzyme, and we finally get our desired product. So it's more than just one enzyme. Sometimes our product will come back here and act on our first enzyme and shut this system down. If we don't need very much product, we'll go shut the system down after we make our first product. And so here's the linear feedback inhibition, and then here is the cyclic feedback inhibition example, where our product goes off of the cycle and we can continue going back to enzyme 1, 2, and 3. And so there's not a huge difference there, just some end with the product being the finished deal, and others will make a product and they'll make a chemical or an organic molecule that also serves as the reactant for the beginning of the reaction, and so they can kind of go on as a cycle. All right, redox reactions are really the basic chemical component of all of our metabolic pathways. We call them redox reactions or oxidation reduction. If you give up electrons, you are oxidized. And if a molecule accepts electrons, it is reduced. Try to remember it like Leo goes Gur. Leo, lose electrons, oxidized. Gur, gain electrons, reduced. And that'll kind of help you keep those two straight. Coenzymes are molecules that can accept other molecules in redox reactions and help them to transfer electrons. And so when we get a whole bunch of these coenzymes together and enzymes together as well, we call these an electron transport chain or an electron transfer chain. You have these in the mitochondria. They're what help us make all these hydrogen ions for powering that ATP synthase, and we have them in plants as well, electron transfer chains and photosynthesis. And what it is really is just a series of enzymes that are all bound in this membrane, and they're going to give up and accept electrons in turn. And those electrons are like the energy source for this reaction to work. And so here's the simplified cartoon of the electron transport chain. And as that energy goes to lower as the electron goes to lower energy levels, we are harnessing little bits of that energy and using them in the electron transport chain so that we can eventually make our products. That's what we use, is electrons to do that job. So cofactors in our metabolic pathways, most of our enzymes, they're not an island. They don't work alone. They need help. They need regulation, and that comes from cofactors and coenzymes. Cofactors are atoms or small molecules that are necessary for enzyme function. Sometimes they can be really small organic molecules, or even like those trace metals that you get in like your daily vitamin that are like zinc and magnesium, and you're like, whoa, I didn't even know we ate stuff like this. Iron, a lot of those little metals can be useful as cofactors. And then there's coenzymes, which are organic cofactors, like a little larger vitamins, and they can become modified during a reaction to help our enzymes out. So enzymes, they need help. They get help from cofactors and coenzymes. So ATP is our example of a coenzyme, and ATP you already know is adenosine triphosphate, and it can help our cells drive endergonic reactions, which you'll recall are reactions that need energy to come into them. And ATP is a nucleotide that has 3 phosphate groups, and it'll transfer a phosphate group and the energy to other molecules, because when you cleave that outermost phosphate group, you release a lot of energy, and they'll use that energy and transfer that to other molecules so they can use that energy. So then phosphorylation is the term we use for a phosphate group transfer, which we just described. And once we go ahead and phosphorylate something else, we create ADP, which is adenosine diphosphate, and he just kind of floats around waiting to get that additional phosphate group phosphorylated to him, and that'll return him back to ATP, and he's a high energy currency for the cell, and he can go around and power other reactions. So we call that the ATP-ADP cycle, because these two are constantly changing back and forth. So here's the cycle for endergonic and exergonic reactions with ATP. So ATP has lots of energy. That's why they make it a little gold coin. He goes and gives up some energy to become adenosine diphosphate and inorganic phosphate. Later on, he gets energy back and gets his phosphate group back, and he becomes ATP, and he can go be used somewhere else. Here we have the same example. We have a reduced coenzyme, so gain electrons reduced. He has more electrons than he needs. He gives up an electron as energy. He becomes oxidized, lose electrons, oxidized, floats around as the oxidized coenzyme, and then as he comes back around to another location or another job, he then gains electrons and energy and becomes reduced again. And so that's kind of how these cofactors work to transfer energy to make these systems work better. So cell membranes, if we take a little closer look at cell membranes, they have a lot to do with our metabolism because they're a great surface for a lot of our reactions, especially our membrane-bound electron transport reactions, to occur on. So our cell membrane is organized as a lipid bilayer. You guys already knew that. and it has many proteins embedded in it and attached to its surfaces. And those phospholipid molecules have two parts, the hydrophilic heads that are interacting with water and the hydrophobic tails that are trying to stay away from water, and they make their barriers for the cell pretty naturally, and I think you guys already have a pretty good grasp on that. So we call this fluid mosaic. That's what we call the plasma membrane of the cells, because the phospholipids themselves aren't necessarily chemically bonded that strongly together. They're just trying to all avoid the water. And so when they try to avoid the water, they just get kind of all bumped together. And that lets them drift and move around more like a fluid. And we call it a mosaic because if you've seen mosaics like in artwork, it's lots of different pictures and pieces of different colors and textures. And the fluid mosaic model for the phospholipid is it's not just phospholipid heads everywhere. It is proteins that stick out of this phospholipid bilayer and carbohydrates and steroids and stuff all over mixed in there. So that's how we get the name mosaic, is because there's a lot going on in a cell membrane. It's not just phospholipids. And so here's the diagram, one layer of our lipids with the hydrophilic heads facing out, and then the bottom layer of our phospholipids with the hydrophilic heads facing towards the interior of the cell. Those hydrophobic tails are hiding from the water, and this gives us a nice fluid mosaic model, and we can throw proteins and carbs all over the place in here. So, why do we add proteins to our plasma membranes? Cell membranes have a function. It's not just protection, but they also have proteins associated with them. Those proteins can communicate with the outside world. They can identify other cells, let the cells know who you are, and they can also transport substances into the cell or out of the cell. Peripheral membrane proteins are pretty much just on the outside, and they'll just temporarily attach to the lipid bilayer's surface by interacting with those lipids or other proteins. But then there's integral membrane proteins, and those are permanently attached in that lipid bilayer. They're generally on, and they generally have ends sticking out kind of to the outside world and to the interior of the cell, and they're really stuck inside that fluid mosaic. And so here's a few of those membrane proteins and kind of what they do. You have adhesion proteins, which help cells stick to one another and to their extracellular matrix, so everybody knows who everybody is. There's a receptor protein, which will initiate change in cells' activity in response to a stimulus. So these proteins kind of stick out into the external world, and proteins or hormones can attach to those receptor proteins and then be taken into the cells. Perhaps a virus can attach to those receptor proteins and be taken into the cells, so they're important for those as well. And then we have enzymes, of course. Enzymes can stick out of plasma membranes, and you guys already know what they do, catalysis. And then we have transport proteins. These are what we're going to talk about a little bit more. Transport proteins function in moving specific ions or molecules across the membrane. Some of them require energy input, some of them do it without energy input. There's a little bit of variation there. So our first example of moving something across a cell membrane is diffusion. Hopefully you're already familiar with this. Diffusion is the movement of molecules down a concentration gradient, which means they're moving from an area of higher concentration to an area of lower concentration. So this is passive. It's not taking ATP or energy to do this. They just do it on their own. So we can use this to move substances into or through cells if the substance is friendly with lipids. If it's got a really big substance or it's got a charge on it, it's not going to be so friendly with lipids, and it's not going to be able to zip right through. So it's got to be able to pass through a lipid bilayer to be able to diffuse readily into and out of cells. Substance diffuses in a direction set by its own concentration gradient. So let's say we have a lot of oxygen outside of the cell and not much oxygen inside the cell. Then that oxygen, which is really small and doesn't carry much of a charge, will just naturally bounce around and diffuse through the cell until we have like approximately the same oxygen outside and oxygen inside. So we've moved down a concentration gradient. Hopefully that kind of makes sense. So diffusion is, let's say we're something that our size and charge don't keep us from moving through our cell membrane, is going to depend on a few different things, how quickly we're able to diffuse. The first thing is size. If you're really, really big molecule, you're not going to diffuse as quickly as a really small molecule like oxygen gas would. Temperature. You increase the temperature, you increase the speed at which molecules vibrate and move. You increase the speed at which molecules vibrate, you increase the rate of diffusion. So a warmer media would have quicker diffusion than a colder media would. How steep the concentration gradient is. If there's twice as much oxygen outside a cell as inside a cell, we will diffuse reasonably quick. If there's 5 times as much oxygen outside of a cell than inside of a cell, we have a much steeper concentration gradient and we are going to have a very fast diffusion occurring. Charge, if we are trying to move through a lipid bilayer and the lipid bilayer is not charged, it's charge neutral, and we have some positively charged sodium trying to get through there, he's not going to diffuse very well. It's just not going to happen. And then finally, pressure. If we can pressurize one side or pressurize the whole system, we can speed up diffusion that way. So hopefully conceptually those kind of make sense to you guys. I'm pretty certain we're going to be doing a lab on this, so you'll get to see this kind of firsthand. But that's where we're going to stop for today, and we'll jump into more metabolism next time. So thank you.